Prathamesh, start
Yes, sir.
recording.
Yes, sir. It is. Done, sir. Done. Thank you, Prathamesh. So, good evening to everyone present here. So, I, Bipras Kumar Tiwari, pleased to welcome our MBSI All India President, Dr. A.M. Deshmukh, sir. We are very fortunate that today with us, Dr. Prakash Salami, sir, is present here. And also, the Dr. Mosmi Devnath, madam, is here.
Thank you.
So our today's topic, roundtable discussion on the very impactful topic that wastewater treatment using the microbial consortia. So let me introduce this topic within 2 or 3 minutes. I will take. So we know that with increasing urbanization, industrialization, and the pressure on the freshwater resources, the effective treatment and the reuse of the wastewater has become a global necessity now. So we have the traditional chemical and physical methods of the treatment, and those are also useful and but often the expensive. They are the energy intensive and the sometimes environmentally damaging. In contrast to the physical and the chemical method, the biological treatment means when we use the diverse microbial consortia, it offers sustainable, cost-effective, and the eco-friendly alternatives. So generally, microbial consortia refer to the mixed communities of the microorganisms, including the bacteria, fungi, protozoa, and algae that work together synergistically to break down the organic and inorganic pollutants present in wastewater. So the different microbes in the community complement each other's function because there are the mixed communities are present and some degrade the complex organic matter, others remove nitrogen or the phosphorus, means the inorganic compound, and still helps in sludge reduction or the odor control. So these microbial communities play a key role in the several types of the biological wastewater treatment system, from conventional activated sludge processes and anaerobic digester to constructed the wetlands and the biofilm reactors. Also in the industrial setup, the customized consortia are now being developed to treat the highly toxic effluents such as the textile dye, pharmaceutical residues, and the heavy metal. And one exciting direction is the development of the engineered consortia where the specific microbial strains are combined to achieve the targeted outcome. So there is a growing interest in the coupling microbial treatment with the resource recovery, and for example, the extracting biogas, nutrients, or even clean water from the process. So use of the microbial consortia also bring its own set of the challenges. So the most of the challenges are the maintaining the stability, optimizing environmental conditions, dealing with microbial competition, and ensuring the consistent performance at large scale. So in today's discussion, we have the opportunity to share our knowledge. We have the opportunity to share the field experiences and our research findings. Okay. So as a researcher, as a faculty members, or as a scientist or a students, I request everyone to share his scientific insight, his means their scientific insight, if the research experience or the field experiences on the topic wastewater treatment using the microbial consortia. So now this means it is open for discussion. So I request anyone, research scholar, faculty member, scientist, to share the research experience. So just I request.
Can I start?
Yes, madam.
Okay. Namaskar, everybody. I'm Dr. Mosmi Devnath. So I work in the field of halophiles, and we have been trying to isolate bacteria from Sambar Lake. Right now, our work in the field of halophiles have increased. We have also worked in the field of textile effluent bacterial isolation. where we got some halotolerant, and now we have moved to another group, that is Andaman mangroves, where we have again got some halotolerant species. Looking to a collection, a consortia has been built, and we work on the field of biosurfactants. So these potential biosurfactant producing bacteria that we have isolated from all these different sites are very potential is wastewater treatment. So we have tried to utilize our biosurfactant producing bacteria along with the extracted biosurfactants for a technique called as moving bed biofilm reactor. These moving bed biofilm reactors are very small sized polypropylene floaters, and these are able to attract the microbial community by forming a sort of a biofilm. And our action with the inducers, that is the different extremophilic, specifically halophilic bacterial species that we have isolated. So these isolates are inducers, and they start colonizing on these biofilms. And these biofilms that are formed on regular basis in the moving water are also shredded off in due course of time, and then new system is again created. So it's an ongoing process. During this process, we found out that if we add our extracted biosurfactants also, the initiation and the sustainability of the biofilm is prolonged. They are able to reduce the pollutants, and a considerable amount of treatment is observed. This whole process has been checked in all the textile effluent colors of water. As you know, textile effluent, every day if you go to any industry, you get different colors. So all these colors are having a specific type of a chemical activity, and these have been worked upon looking to two basic domains in India. Sorry, I stay in Jaipur. So Jaipur belt has a wonderful textile belt, and that is the Sanganer. So Sanganer had around 2,000 textile industries. So we used to procure water from there. Although we have also worked with some organic based dyes also, like we have worked with indigo. But indigo, because I mentioned, because most of our jeans are having that color, and there are big industries only working with indigo. But we have also worked with different other dyes apart from the azo dye, which is the major constituent of the Sanganeri prints. Bagru is another sector in Rajasthan, Jaipur specifically, where we also have a lot of they are usually hand printed textiles that they have. So they have vegetable dyes also. So our treatment is working excellent in all these decolorization. Not only decolorization, the metal content is also reduced. Secondly, we have also used this type of a technique with the ceramic industry. Ceramic doesn't have an organic pollutant. It has an inorganic pollutant. So our major thrust was how to give them a water which they can recycle it. So we achieved that point. And the third thing where we have already used it is in the sewage treatment plants. So this technique is ongoing and is used by many people around the world. They do not use this consortia that we use. They do not use the biosurfactant. So the use of these two things is very novel, as it instigates the process. It's in a very We now don't need a complete set of wastewater treatment plant. We don't need flocculation coagulation. We can work with a single aerator system, and it gives them a better ability to overcome the huge cost of electricity which any industry faces. So the space and the electricity, we can reduce the cost in a good amount. Thereby, the use of consortia that we have tried to make can be benefited for the betterment of the country. Any questions? I'm ready to answer.
Thank you so much, madam, for sharing your research experience.
Thank you.
Research scholars, if you have any query to madam, you may ask now. Okay? And if you cannot unmute yourself, you can write in the chat box. I can read out it to madam.
Yeah.
Any students or the researchers or the faculty members, if you want to share. Because this one is the platform where you can share our knowledge, or we can also discuss if you have any problem with the expert present here.
Yes, I think so. There is one hand. Please speak out.
So, Pouran.
Yeah, please.
Unmute yourself.
Yes, please unmute.
Pouran, you can unmute yourself. Otherwise, you can write your question in chat box.
Yeah, good evening, everyone. And I just wanted to ask about how much the consortia method is feasible in large-scale treatment. Thank you.
Yeah, very good question. Very true. Whenever you have a consortia, there is always a either it's an antagonistic or it's a synergistic type of a reaction that the bacteria faces. So what we have done is we usually go for a continuous evaluation of this consortia. So we use some of the latest techniques to understand whether actually our consortia is working or not. So this whole system is based on a upgradation method. For example, if I want to go for 3 KLD or 4 KLD of water, I don't use that many number of amount of the microorganisms or my biosurfactant. So we usually go for a scale up. When we go for a scale up, our work is only to induce it. So when we prepare a consortia, we usually go for taking the biomass and then check it through the different types of techniques. We usually go also for the confocal microscopy and check actually where are these particular bacteria, what is the number of the maximum colonizers. So we also go for certain phase contrast and this fluorescent microscope together. We try to find out the number of cells there. And then we also look forward for the SEM analysis, and then we try to look. So what we basically try to do is we try to look into how many bacteria and what is the constituent that goes with particular timing. We also look for certain types of activity which each of the bacteria is trying to do. It might so happen that all those bacteria are not having the same type of growth in due course of time. So initially we screen them that they are almost more or less of the same growing pattern that they have. And in due course of time, they don't have such antagonism among each other. So the consortia that we have made is having a certain constituents of our biosurfactant producers also. So we basically work with two types of biosurfactants. One is surfactant type of a biosurfactant, and the next one is a rhamnolipid. So we try to keep those bacteria which are producing these constituents in a certain number, a certain ratio, and we maintain it, and then we go ahead. Also, we try to look every time on the plate whether that particular type of before you found this consortia, whether there is an antagonism reaction happening. Many a times it so happens that one of the bacteria grows, it overgrows, and then you don't have the total colonizers growing together. So once you consider all this point, then you can make your own consortia. But in our case, our major formulation is to form that extracellular mass that they are continuously creating. So our bacteria produces extracellularly this biosurfactant. So our basic work is to make the consortia for the certain constituent of the biosurfactant that would be effective in reducing the pollutants.
Thank you so much, sir.
Thank you.
Wonderful, ma'am. Thank you so much, ma'am. I have been in this research, wastewater treatment using microbial fuel cells for quite a while, and
Wonderful.
I found that it's very interesting about your research on using the consortia. But I have one small doubt again, which is the dynamics of the microbial communities changes in course of time and it stabilizes. Does it interfere very much in the constitution you are adding to the thing? And what type of harboring metal do you use for treating while treating it in the reactor, ma'am?
Sir, I had been the finalist of one of the very good funding agency, IGSTC. So I was called to Bonn on the 3rd stage, and they mentioned the most important metal is lead. Okay, so I particularly look forward for certain bacteria, if they can resist some of the amount of lead, then they can be sustainable. Okay, and usually in all my textile effluents, I do have lead. So these are allowed to work also on the lead containing solvent. So here my effluent contains lead, chromium, cadmium, nickel, arsenic, mercury, all of them are there. Copper is huge amount. So all these metals, these bacteria are able to sustain. They grow well. So they work well.
Thank you so much. Thank you so much, ma'am.
Yeah.
Thank you, everyone.
Yeah. Thank you so much, sir, for your questions.
You're welcome, ma'am. I actually take this moment to thank Dr. Desmukh sir, because I met him here at Chennai, and it's a very great moment. Thank you, sir, and thank you, everyone.
Thank you, everyone. So now,
so thank you, Viprant sir, for giving me this opportunity to speak. Thank you so much.
Be here with us, madam, for 1 hour, because there may be the question from the students and researchers.
Yeah, sure. Yeah. I'm here. I'll switch off the video, right? I'm here.
Now, any faculty researcher or the research scholars, if you want to share your research experience, please unmute yourself. Anuja Gangurde, Dr. J. Usarani, Dr. Prasun Kumar, Dr. Aparna Farande, Dr. Aurula Jagan, Dr. Prakash Salami, am I audible to you, sir? Can you hear me, sir?
Yeah, I can hear you.
Now we would like to hear from you, sir, on this topic, sir. Your research insights on this topic.
Yeah, give me some 5-10 minutes.
Sure, sir.
Let us explore some other experts also.
Dr. Prasun Kumar, sir.
Yes, sir.
Kindly share your scientific.
Hello.
Yes, sir. You are audible, sir.
Yes, sir. So, thank you so much, sir. Actually, to ma'am, I wanted to ask a few things regarding the experimental part. Like when we are going for microscopic observation of these consortia, how can we distinguish between different organisms or their level of growth?
So this question is to Mosami madam.
Yes,
Okay.
yes, yes, sir.
Okay. So, sir, we have looked into the confocal. I think so. You know their attachments. So when you go for a phase contrast microscope, you can see all the outline, right?
Yes,
So if I look into the outline of the bacteria, I can easily make a difference between a few types of bacteria, right? Done.
yes,
So what we do is, you can do the same thing in case of SEM also. So we do have in our university FESEM, and we have also looked into the confocal microscope. So both of them, so the SEM will give you the external
surface view. Yes,
topology. Okay,
yes.
so the surface topology can be looked into. So if I look into the surface topology, I know that if I have a picture of all the separate bacteria, and then if I compare it with my consortia, I can literally make it out.
Oh, that will happen, ma'am, when they are morphologically distinct.
Yeah, but yes, very true. There are certain which look same.
Yes, and then
Yeah,
identifying their number will be like tricky. And also,
yes,
yes,
we do have such issues also, sir. So with less number of bacteria, it's easier. When the number of bacteria is something around 16, 17 that we usually use, so it becomes a bit tricky at times. But if you use both the systems parallelly, like you use a confocal also and you use a SEM also, then it becomes a bit easier. It's not so easy, but it becomes a bit easier. Now, in our case, sir, we are just using it to trigger the work. So they need to form that consortia. And then once the consortia is formed and they are growing very well, so what we do is we induce this molecule to form the biofilm. So the biofilm is not necessarily made by our consortia. So there are so many other organisms also inside the water which starts colonizing. So our work is to reduce the timing required for the biofilm formation. So in
okay,
case, when I was there in that particular conference and what you call it, the project meeting. So my counterpart there from Germany, they already had certain sewage treatment plants where MBBR was being used. Even in my campus also here, we do have it's a 50 MLD sewage treatment plant, and there they are using this. Now they use just the floater, so they are using it where to instigate the process, to start the process they need around 1.5 months minimum. But when I use it in my form of consortia with the biosurfactants, the number reduces. So accordingly 45 days is required, so I can achieve that in 25 to 30 days. So I reduce the timing, and because there's a narrator there at the bottom, so water needs to again and again, it has to, so they need certain soluble oxygen inside it, so at least a little amount of energy is being utilized, but then you save that amount. So for all MBBRs you need a narrator. So that energy that you are saving is worthwhile for an industry. And
correct.
secondly, there should be certain amount of foaming. Now, in case all these type of microorganisms colonize, what happens is there is a requirement. For example, Surat, I got a call. They had a CTP, and it is all related to textiles. Now they continuously were using MBBR, but the amount of foaming which was caused, that was even at times sometimes very, very difficult for them to handle. Now, if you use certain molecules like this, though, what would happen? They would interact with each other, and that foaming part is always under control. Although rhamnolipid is a very good foaming agent, but what we are doing is we are trying to make a certain ratio, and that ratio is promoting the reduction of pollutants rather than making the system a little hazardous or uncontrollable.
Mhm. So when we are doing, sorry to interrupt, ma'am. Yes, you may go ahead,
Yeah.
please.
No, I'm okay. You can
Okay.
ask me question.
Just actually, so regarding same, so we need to dilute our sample before doing same, because if they are forming biofilm, then biofilm layer will cover the cells.
Can you repeat your question, please? Sorry.
Ma'am, for same, yes, no problem.
Sam, you
So for
don't do
same
that
analysis,
dilution, sir. That is the reason why I asked you to repeat. When you do
uh,
SEM, sir, what you do is you make your consortia and then
mhm,
you just centrifuge it, put the cells down, and you take the biomass 72 hours. Okay.
okay,
So that
okay,
is the crisp dry, because in FESCM, anything which has water, the person won't allow you to run the sample. So
yes,
you
yes,
have
because
very
of the
crisp,
vacuum.
and then you have to coat it. Na? The coating has to be done with gold. So you can't do it. So once your cells are just layered on that particular grade, then that is how it goes. Okay.
And how about confocal microscopy, ma'am?
Confocal, you
Confocal, how to
find
prepare the
out
sample, basically.
yeah, so you can put it on a cell, a slide, and there is a whole technique, sir. You have to just do the same thing, and then you can look into, because we work with both the molecules. We work intercellular and intra. Basically, it's an intracellular molecule. Also, we also work with the pH globules. So inside that also we want to look, and the same bacteria produces the what you call it, the biosurfactants. So when we go for outside material, so you can basically, we don't look into biosurfactant production in that case. We go for EPS calculation, exopolysaccharide calculations. But when we look into the cell structure, so the cell structure in both the cases, when you look into FESCM structure and when you look into confocal, you can actually look into the microbial diversity present there. And then if you do the same thing with single cultures, then you just make a comparison. That's what we do.
Okay,
Yeah. One
okay,
of our
okay.
papers has been communicated in this area only, where we have done this morphological distinction. Once it comes, then I might be able to share. Okay.
Sure, ma'am. Yes, I would be waiting for that. Actually, recently I tried to do confocal for the first time.
Uh-huh.
So for a biofilm forming strain, but I
Uh,
was struggling with how to keep them in a slide because we need to do
it's too much,
staining
and
and
there are
staining has
lots of
to.
it. Don't do like
Yes,
that, sir. When you make the consortia, you start working at that level. Don't go for that biofilm level, because there
okay.
millions of microorganisms are there. It's not just bacteria. Get millions of microorganisms, because all those microorganisms will come and eat the pollutants. No? So there's so
Mm-hmm.
many things. It's not just the bacteria alone.
Okay.
I hope
And
it's convincing.
yes,
Yeah.
yes. And because you said PHA, so I got a bit more curious. How much amount of PHA is there in your consortia? Have you calculated it or checked it
Yeah.
anytime?
Yeah, we did get, I think so that would be in a different forum.
Okay, okay.
A call kursi. Okay.
Okay, ma'am. Thank you so much. Yeah.
Yes.
Thank you, Prashant sir and Mosmi madam. So, madam,
Yeah.
one question in the chat box. I think
Haha.
question that can we prepare consortia for electronic waste degradation, and what time required for the degradation?
I've not tried, sir. But yes, it would
Madam, I
be
have a
really
question,
nice.
if I can ask.
Yes, sir.
Hello.
Yes, Atmik, you can ask.
Madam, if we are going for the using the consortium for the dyes degradation purpose, basically reactive dyes, we can say it out. So
Yeah.
basically, if I am using the consortium for the scale up purpose, how we can
Yeah.
prepare that scale up proportion? Because as for the identification purpose, I have already gone for the metagenomic study, so we can identify it. But the main reason behind that is if you are go for the aerobic degradation purpose, बराबर से we are continuously mixing that, then the degradations proportions will be decreased. But when we go with the anaerobic conditions, बराबर, then the dyes will be break down, and the uh, product that will be generated will be cyclic.
Sorry for the call. Yeah, please tell me.
Yeah, madam. If we are going for the anaerobic or microaerophilic degradation purpose, then it will go with the cyclic compound formulations. So
Yeah.
basically, we need the breakdown that in a way that will be not be toxic in the form of that. So
Okay, so how do you see breakdown is happening?
basically, the enzyme will be released or as a reductase and
No,
whatever that.
if you want to look into it, so the best thing is go for FTIRs. So you will see the
No,
intermediate
that
compounds.
I have already all the purpose is being studied for that purpose. Okay, how it is breakdown and the pathway is all being ready out. But the
Okay.
purpose is when we are going for the scale up purpose,
Uh-huh.
and considering both the anaerobic and anaerobic or we can say it as a microaerophilic conditions and aerobic condition that both being required in that scale up purpose. Otherwise, if we consider only the aerobic conditions, then the breakdown percentage will be decreased. And if we go for the anaerobic condition or a microaerophilic condition, then the cyclic compound will be increased. That will be more toxic than the reactive days purpose. So if we are going for that purpose only, then how we can scale up that portions?
See, the first point of using a consortia is the consortia which you are conveniently working and handling properly. When we start, as I told you, we have isolated bacteria from different types of extremophilic regions. So my first thing which I do is, how do I make a suitable consortia? And it again depends on the type of the pollutants. For example, I specifically showed you 3 instances where we are using a consortia, where we are, for each of the type of pollutants, we have a different types of consortia. Now, what you said is you have 2 different conditions in which your microorganisms are growing, aerobic and anaerobic both. How can you handle it? There'll be a lot of confusions between your microorganisms.
Basically, that microaerophilic conditions that organisms both that grows out. Basically, when I
There
take
will be
the
a lot
dyes
of
and
antagonism at certain points, sir. If you just
yeah,
check them, it is very difficult to grow. It
but
can be sustainable for 24 hours, 48 hours, and even till 72 hours. But after
but
that,
madam,
they'll form a chaos.
I am maintaining it from the last 3 years,
Oh,
basically.
then so sir, you have the answers.
Yeah, but main problem what I am facing is during the scale up purpose because when we just go with the high nutrients and amount and all that, that will be increase the cost. We decrease the amount of the nutritions and just go
Scale
for
up.
the non-stearic condition, then the breakdown that contain have the high amount of the cyclic compounds. And when just
Yeah.
go for the aerobic condition, then it will be the percentage of breakdown being decreased out. So
You're
we
very
can't
correct, sir.
manage it. For
It's
the
very
purpose.
difficult. Your consortia is a bit
Yeah,
tricky. It's a
because
naughty
individual
compo.
organisms is doing the same thing.
Yes, sir.
Individual organism is doing the same
You
thing. Then
can do
basically,
one thing, sir. You can do one thing. You can separate both the consortia, okay, and give them part by part treatment. Okay,
yeah,
like
that we
you
can
give
do.
them 7 days, one type of a treatment, and 7 days, because finally you need it. Your output should
Clean
be like,
water, yeah,
yeah,
clean water.
you can make this type of a consortium, because initially when you dose your treatment, this because your consortia is a bit naughty group, they will fight
Yeah,
for sure. After 72 hours, by 120 hours, they will all go mad. What I personally mean.
yeah.
So what you can do is, sir, you differentiate it into 2 groups and give them 7 days treatment, one group 7 days treatment. So your pollutant level in all the cases will form intermediates and they'll settle down.
But that's the major issue with that combination because I am using for the mangroves ecosystem consortiums. I do not have separated out because when you go for the separation of that particular consortium, nature consortiums, then the most of the organism will not be grow out.
You have to find out a solution, sir. Our consortia, we make it. We design the consortia according to pollutants. So this
But
consortia is totally in our hand and it's totally novel. But if
Yeah.
you have a consortia like that, then you have to take out, because if you can distinguish two different types and then go for a treatment, then it will become better. I personally feel, because then you can attack certain group of pollutants and the next group you attack. Because if you use them both together, there will be certain group which will diminish, which will be
And
diminished
then
in
one
number,
will dominate. Yeah.
and one will dominate.
Yeah. Sure. Thank you, ma'am.
Thank you.
Thank you, Madam. Hello, Anjali Patil.
Hello, ma'am. My question is related to the electronic waste degradation, and it is my PhD topic.
Okay.
But some experts say that
I have
electronic
not
waste.
worked, sir, ma'am. I have not worked in electronic waste, so I don't
Okay,
have it.
okay. Okay, fine.
Yeah, I have not worked.
Okay, okay, ma'am. Okay.
Dr. J. Usarani, Dr. Biswas, Dr. Aparna Farande.
Sir, I worked on another part of wastewater treatment, that is on bioenzyme. So I thought I'll get some inputs. That's why I attended. But it is useful. Even I am working on the biofilm. We prepared. We have gone for SEM and TEM, but I could not preserve it properly. So still work is going on. Yeah.
Okay, thank you.
Thank you, sir.
Dr. Biswas, Dr. Aparna Farande, Dr. Anuradha Gun, Dr. Led Atmik. Dr. Led
Sir, I
Atmik,
have already asked. Yeah, I have already asked.
you may share your research experience. You want to share your research with the audience, means participant. Because you have a huge experience in this field, so you may share your research experience. Hello.
Hello, Doctor. Good evening. This is Dr. Arul here.
Good evening.
Hello. Can you hear me?
Yes, sir, you are audible.
Yeah, this is Dr. Arul here. Yeah, okay, fine. Great. Okay, we have been working on halophils, thermophils, and alkalophils as well as acidophils. So we worked with mostly with consortia, which is enriched from the contaminated sites. So I think there was one question regarding identification or distinction of bacterial profile. It is highly recommended because nowadays regarding the publication, they are asking for NGS, next generation sequencing technique, and go for that. And then you can go for the bacterial profiling and percentage, how much amount of percentage of bacterial dominance is there. Okay, this is one point, and one more is regarding this halophils and the usage of this consortium in different wastewaters like tannery, cosmetics. We have to also focus on the heavy metals. So, for example, chromium 6 be converted to chromium 3, something like that. And apart from that, petroleum hydrocarbons. So we worked on this along with the petroleum industries. So we collected the refinery wastewater, and under saline condition, we try to treat them. And our consortia sustained up to 180 grams per liter of NaCl concentration. And apart from that, we also focused on thermophils, which we have already discussed in our previous session. So in that, I have intimated that it was able to survive up to 60 degree. And acidophils we got from mining industries, so which was able to treat the cosmetic industrial wastewater, and it was also under acidic condition. It was also able to degrade the pHs, and the pH we used is at 4 three, four, something like that we used, and we try to treat the organic pollutants. So best way to treat the industrial wastewater is enrichment of consortium from the contaminated sites, because we have to focus on where exactly this kind of bacteria will be available. So we go for the brine water from desalination industries, and these are some of the sources I'm pointing out. And if some questions, we also work on this microbial fuel cells. When using halophils, we try to treat the aquaculture wastewater. So if anything, we can discuss. Thank you.
Thank you so much, sir, for sharing your research experience. So now, students, Tansia, Saxania, Tanisha, Kika, if you have any question or if you want, if you have any doubt, you may ask here. Tanisha, Saxania.
Excuse me, sir. I have a
Yes.
small question. Yeah.
Sure,
In
sure, please
Consortia, what
go ahead.
all organisms are there? We need to know that identification of the organism is also important. Is it important, or we can go with the
Yes.
natural process?
No, it is very important. Now, if you are going for publication or any kind of patent, you have to give what are the bacteria present in your consortia. And when you are operating a reactor and at different organic loads, you have to take the samples. Again, you have to go for bacterial profiling. The percentage of dominance has to be reported, because most of the reviewers recommend for that.
Thank you, sir.
Thank
You're
you.
welcome, ma'am.
Arul sir,
Yes, Dr.
hello.
Yes, please.
Yes, sir.
Please.
I'm glad to see you on this platform. I have been following your research work since long, and I have been citing also. Great to
Oh,
see you here also. Yes, sir. Just two questions that I wanted to discuss with those who are working in this area. No? So one thing
sure.
was
Please.
this NGS thing that you do. Uh,
Yeah.
so in India, if we are doing it, what would be the cost, and is there any company that provides the solution for means consortia identification? No? If we give mixed culture, they will give us the profile, uh, the the community
First
analysis.
thing, okay. First thing I want to tell you is I'm based in Saudi Arabia. Okay.
Okay. Uh-huh.
So I think we send it to either to Germany or France, because we have a strong tie-up with them. And then apart from that, we find some of the companies which can do at low cost if you are regularly sending the environmental samples in Korea as well as in Singapore, something like that. So we try to collaborate, or we try to do this analysis. But after sending the articles, the reviewers strongly recommend what is the percentage, what is the novelty, how far it is going into this. So even if we say that we have identified this, they ask what is the percentage of dominance in at this particular OLR organic loading, something like that.
Yes.
There are some fishy questions are there we have to address. So in that case, the research is going little bit advanced, and then we have to focus on that also.
Mm-hmm. And regarding sir microbial fuel cell, do you think whenever we read about it, at least it is my opinion that whenever I read, I am unable to relate the amount of electricity being generated and whether it is useful at the end or not, means the amount of electricity that is being generated. So if we are integrating two process, suppose a kind of fermentation, and then the effluent is going towards MFC, and then finally to maybe anaerobic digestion. These three steps are combined together. So the amount of electricity produced, is it enough to reduce the cost of the overall process or not?
Regarding MFC, I cannot directly say it depends upon certain single features or something. There are multiple things we have to focus. First thing is the electrodes, cost of the electrode materials. We have to focus on that, and it should be eco-friendly, something like that, because carbon-based materials are used nowadays used. And then apart from that, what is the bacterial consortium you are enriching, and then you try to treat that. And regarding your question, the electricity produced can be, it depends upon, you will run at a different organic loading, and particular organic load may give you the amount of I think the high amount of electricity, but that you have to correlate with the cost, how much you have spent. Everything has to be accounted. So this is not only just stopped with certain things. We have to focus on many things, because when it is going into laboratory, apart from laboratory, pilot scale, or into TT, technology transfer, or something like that, then we have to focus many things on this MFC fuel technology. But nowadays we are much focusing on this biohydrogen production. So I think I recommend mostly on this hydrogen production from MFC also.
Okay, but sir, the yield, the amount of hydrogen in MFC, the compared to the dark fermentation. So dark fermentation will give us at least 4 mole per mole of hydrogen. Here the yield may be low, no?
Here you are doing dual things. It means one side you are treating the wastewater, and you are
Okay,
removing the
okay,
COD
sideways
content. And
generating.
here the sludge generation is very less compared to the other things, and you are getting the electricity output also. So
Okay,
so you have to focus on both the things. It's
okay,
little bit tricky, but still you can come out. And one more thing is the design of the reactor and other things also we have to focus.
okay. So you have tried. To what scale have you studied, sir? As of now, Miss lab scale,
Currently,
small reactors, or
regarding this MFC only with a lab scale we have done, but regarding this petroleum hydrocarbons, we have gone into this pilot scale studies. We try to implement this CSTR or a secondary treatment plant, whatever they are having. We go there and we try to provide our enriched consortia, and they try to mass cultivate, and they try to employ, and they found some good results. But whatever you are getting in the lab, you will not get the same results over there. There are some complications also when you are go into the field. It is totally different, because in the laboratory you might get 90%, but when you go into the field, it is totally different. Almost you will get 70% or 10 to 20%, or even 20% or 30% results will be reduced. When you go into the field, you have to focus many things there, and you need an environmental engineer along with you, what gone wrong, and you have to focus on this HRT, oil. So many things we have to focus when you go into the field.
okay. Masimem, are you there?
Please.
Sorry, I couldn't get you.
Yes, sir. No, Arun sir, in continuation to what you have said, na, I wanted Mausami ma'am opinion also in the same context, because she may have also done some experiment in the
Yeah.
lab and then scaled it up.
Okay.
So whether she also faced similar kind of issue, or the conversion was as expected compared to the lab.
Sure.
No, we
Yeah.
have gone till 3 KLD only, and we got very good results. So in that way, the scale up was our major challenge. How do you scale up? So we tried to optimize our scale up in such a way that looking into the counts, we kept on going according to our scheduled plan, and it worked well. So you just can't change your whole process when you're going for an industrial level. So you need to plan it and first make all the basic things according to your pilot scale that you have tried. So we tried with a pilot scale initially, and then we scaled it up to the same level. So we basically go only for 1 liter of our sample initially, because we use liquid samples at that time, and we use a crude biosurfactant. So in that case, our system works very well. With that certain amount, we keep on acclimatizing the polluted water and try to grow it in that. And then we mix it with the bigger volume of water. So it doesn't go in a very different manner, but it goes according to what we thought would have gone. So as because our basic matrix differs, I've worked with some of the companies where they are working with different herbal components also. And we also got water which contained marigold flowers, their extract. So that was absolutely yellowish, or you can say saffron color. So, and it had lot of very strong, pungent, organic, what you call it, they were a lot of solvents there. So those were used for extraction of the biomolecules from that marigold flowers. So that also we tried to use it, and our consortia worked well. At that time, we had a mix of 16 bacteria, because the number of components in that, and especially that stinging smell, which was the cause of that industry to be closed by the government, was our major challenge. So we used a very high potential bacterial group, and that actually helped me to go from first stage to second stage to third stage of the IGSTC challenge. And literally it worked very well. And as sir also said, Azarul sir, that this method that we use creates the least amount of sludge. So this consortia, choosing a consortia is selection of the material is the most important thing. And sir, if you are really inclined to go for metagenomics, BioCart may be a good help. I have taken their services. They do work on this, and we have done the metagenomics too in India. They are centered in Bangalore. We used to send our samples right from here by postage. You can also try it.
Okay, ma'am, cost-wise, what is the cost for one sample?
I can give the number to sir. You can directly talk to them. Okay.
Okay, ma'am.
I'll put it. I can communicate to sir. I'll give it to Deshmukh sir, and you can take it from him. So it is like you have to look into what you want. Want to do only
Bacteria
16S,
or
or
both?
you have to do the ITS also. Yeah. So if you go for, like, we went for both ITS as well as 16S. So you have to look into what is your need, what do you feel that your microbial diversity, which way it is, because the community would have both things, and you get a huge data, you have to interpret it and then work on it. I think so. Deshmukh sir has seen me presenting once with that whole metagenomics data when I was in one of the conferences. So we had given it in one of our matrices that we have done.
Okay. Thank you so much.
Thank you so much, madam.
Thank you.
Dr. Prakash, can you hear me, sir? Hello.
Can you please continue this time?
So now
from next time I will join.
So I have shared one link, feedback link in the chat box. I request each participant to fill out that form.
Excuse me, Vikrant sir.
Yes, sir.
So I have marked a comment because that link is leading to the page for virology on
No,
again.
that will work, sir. Huh?
Okay, sir. So we should fill this same form.
Yes.
Okay, sir. Thank you.
Dr. Aparna Farande, madam.
Yes, sir. I had a question like whatever Consortia madam has developed, whether it is available for transfer or I mean, or it's for their own use, like.
Presently, ma'am, we are not selling it. We are not sharing it also. Yeah, it's a customized one. We have already patented it. It's already published patent. One of it is already published, which is working for all of these different types of industries. But now we are already, you can find some of our papers also on MBBR.
Okay.
And these consortia are major patch-up is for the biosurfactants. So we do work lots with biosurfactants where we categorize them. So our bacteria are unique because they are extremophiles. Our bacteria are unique because they are producing biosurfactants. At the same time, they are very good PHA producers, and they are also working simultaneously. When we do the PHA, we make biohydrogen also. So they are like a very good machineries doing all the biomolecules together. So we do customize our group for the type of, we first analyze the water and look into what are the metals present there. So we do a lot of in silico also before we try to make our consortia. And then we try it on a small quantum of water. And if it works well, we go ahead with it. Because, ma'am, if you look into wastewater, it is very different. I still remember in one of my interviews that I had with the DST, so they said that if you're working for textile effluent, it is not fully that you can use your halophile. So I had one of the imprint project also. So in that, what we did was, it was at Telangana, at Warangal, we used that on-site bacteria. So we do have a collection of textile effluent, contaminated soil and water, isolated bacteria. And for certain recalcitrant type of a dye containing huge amount of metal, one color is mixed with another color. That's a very big problem in case of textile. That color breaking, if you look and you continuously treat also, it's difficult at times. Moreover, when these people are putting the colors, they also use certain amount of oil. So that is the thing which is working with our biosurfactants, because we have done degradation of the oil. So what our bacteria does with oil is, first tries to do is recovery of the oil in the initial first stages. So for a couple of hours, say for 24 hours, we have checked with GC as well as with FTR. So they take it and then they start degrading. So we get the intermediates later on. And these textile effluents have all these things of pollutants. They have color, they have metals, they have oil. So all of them has to be broken down. So I personally feel is the nutraceutical, the food color and the textile are the most important. But apart from that, ma'am, in case of human, whenever you go for sewage, there you also have the nanopollutants. So that is another challenge, because with our exudates, the antibiotic residuals are the biggest challenges, which is in the nano form. That is where our bacteria is also working well. So those nano formulated structures which are working as the nanopollutants, it can break down. So this is something that we got with our research.
Okay, ma'am. Ma'am, then currently you are like working like with the textile industry only, like?
No, ma'am, we are working with all different types of exudates. One small system in our university we have already, it's working for the sewage also. We have worked with omni-active industries for this nutraceutical industry it is. For the wastewater, we used another type of a consortia. For the Sanganeri belt as well as the Bagru belt, and we have worked with Prime Textiles in Warangal for the textile industries. We have worked with Dilip Potteries for the ceramic industries. We're trying to use it as best as we can, and we try to get the results, and we try to scale up. And for every scale up, it's a different story, basically.
Okay, so if there's any requirement from our end, I mean, will your organization will be able to take it up?
Yes, ma'am. We will try to do it.
Because I'm an environmental consultant and also have a laboratory.
Sure, ma'am.
Environmental.
Yes, surely. We can try our level best, because that is the reason why I feel is our researchers should not be restricted only to labs. It should go out to the society and serve the society. So that's what we intend to do.
Right now, I have a lot of challenges with the effluent treatment plants. So,
Yes.
I mean, for our various customers. So even I isolate organisms from their ETPs.
Yeah, that
And
is
I'm
the
working
thing
on
which,
those.
ma'am, I was told by one of the DST expert. You can find our work on the DST SIR portal also. You can open it. It is there. So our DST imprint project is already there on it, on the portal. Ma'am, the most important thing that they pointed out is I work with Sanganer, belt, these molecules, this microorganism. They specifically said that when you're working at Warangal, try to isolate from there. So it is always the niche environment which works well, and their microorganism would be the best adapted for that type of a pollutant. So although that whole process is so huge to isolate a good consortia, but it did work well. So we do have two categories of textile effluent contaminated microorganisms too, with two of my PhD students. And we have also tried to mix them together, because our more importance is not going for high biomass, but our thing is going for high biomolecule. So we need to understand that it is not the biomass which is going to absorb or bioaccumulate, whatever the process is. Our system is to break down to a non-toxic form. When it comes to a non-toxic form, absolutely that is what we want.
Yes, ma'am. Yes, thank you so much, ma'am, for sharing your knowledge. Yeah.
Thank you so much, madam.
Thank you.
We come to the end of today's discussion.
Mostly, ma'am, yes, sorry, sir, to interrupt you. I also want to thank you. It was so informative session. I wanted to hear more from you actually. And Vipran sir, if possible, please ask Mosmi ma'am one more session. Maybe whatever suitable dates she may give us the time.
Please, Mosmi madam, to be present in other discussions.
Yes,
I'm
sir.
a part of MBSI, sir. You can contact me anytime. Okay.
Thank you.
Thank you so much.
So thank you all for your valuable time, insightful contribution, and my special thanks to our expert today present here, Dr. Mosmi Devnath madam, Dr. Prasoon sir, Dr. Arula Jagan, Dr. J. Usharini for the contribution, Dr. Aparna Fernande madam for the interaction and participation in the today's roundtable discussion. So the diverse perspectives shared, I believe that truly enriched our understanding of the topic and opened the new avenue for thought and the collaboration. So let us continue to engage, exchange, and evolve together, because this the objective of this means roundtable discussion is to share the knowledge with everyone so that we can grow together and we can grow as a community. So looking forward to our next session with equal energy and the deeper curiosity. Thanks to all for your participation and contribution in the today roundtable discussion. So I request everyone, means especially the means contributor, Dr. Mosmi Madam, Dr. Prasun Sir, Dr. Arula Jagan, Dr. J.U. Sarani, and the Dr. Aparna Madam to kindly fill out that form so that we can issue the e-certificate to your mail ID for your contribution.
Thank you so much, sir.
Thank you so much, Madam. Also,
Thank you.
thank you to our Dr. Prakash Salami sir for being 1 hour with us here. Thank you so much, sir.
Thank you. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.